We saw that we have 20 different amino acids, therefore you can imagine we have 20 different pathways to degrade the amino acids, okay? And amino acid degradation is a very important topic because our daily diet contains a lot of proteins, okay? So in amino acid degradation, very quickly, again, there are many different pathways, and for one given particular amino acid, you may have multiple pathways, okay? And these pathways differ in the organism they appear in, the conditions that they appear in, and the resulting central metabolites that they end up producing. Okay, so these are the differences. Now, for our purpose, I want you guys to understand some amino acids are glucogenic and some amino acids are ketogenic. Okay? And glucogenic means that these amino acids can get turned into pyruvate. Okay? Essentially can go into glycolysis, whereas ketogenic essentially means that it can only go to lipid and ketone body. Okay, so this we talked about on Friday last week. Okay, now, so we went through a bunch of different pathways. For example, those that can degrade in oxaloacetate are essentially aspartate and asparagine. Okay, so you have to understand, or you have to be able to demonstrate that you know oxaloacetate, aspartate, asparagine. Okay, this you have to know, right? Because the structure is very similar. Okay, so this you have to know. Okay, so anything that have similar structures, you should know. Okay, so this you have to know. Okay, now the amino acids degrading to pyruvate. You got alanine, cysteine, serine, glycine, and threonine. Okay. Now, in particular, alanine to pyruvate, this you have to know. Okay, it's transamination, right? So for the part that we're learning about nitrogen metabolism, my emphasis for you guys are on the simple reactions. Okay, so for example, oxidation, reduction, transamination, decarboxylation, hydroxylation, these type of things, right? So as we go through amino acid degradation and biosynthesis, there are just way too many reactions and pathways that I don't think you guys can memorize, okay? So I'm going to break it down into classes of reactions that you should be able to figure out, okay? And on the exam, I will give you, for example, a substrate and a product, and maybe it's 2 enzymatic steps. You should tell me what those steps are. 听懂我意思吗 ？OK， 所以给你一个 substrate， 给你一个 product， 然后中间可能是2步的 oxidation， 或者是1步的 transamination 加一个 oxidation， 那种类型的 question， 你要可以answer， 因为毕竟是一个 chemistry class. Alright, so we also talked about THF and folate. Okay, and alpha Kg related amino acids and Acetyl-CoA. We also talked about that. Okay, and then we also talked about how in branch chain amino acids, the degradation essentially has 3 stages. The first stage is a transamination. You convert the amino acid into keto acid. Okay, so usually transamination is probably going to be the first step in amino acid degradation. You get rid of the ammonia first, and then you're left with only carbon, hydrogen, oxygen, which then is easy to degrade into central metabolites. Okay? 懂意思哈? Right? And then in the branch chain amino acids, after you do transamination, you get these guys that are alpha keto acids. Okay? And in a way, they're just like pyruvate, okay, such that they can undergo an oxidative decarboxylation. You get rid of this carboxylate, becomes CO2, and then you end up with CoA. Okay, so this guy is Acetyl-CoA. Okay, so just like pyruvate dehydrogenase complex. And then once you got Acetyl-CoA, you can then do a process very similar to the beta oxidation pathway, where you're going to oxidize the beta carbon into, for example, here's alkene, here's the hydroxyl, here is the keto. Okay, so that is that, right? And then we started talking about methionine. Okay, methionine degradation. Methionine degradation, it's interesting in a way that methionine degradation occurs through a methyl donation. Okay, so methionine is right here. Methionine first get converted into SAM. Okay, and SAM can then donate the methyl group onto an acceptor. Okay, so here's a methyl group onto an acceptor, such that you become SAH. Okay, so S-adenosine homocysteine. Right, and this SAH can then become cysteine. Okay, so you go from methionine to cysteine, and then cysteine can go to where? Pyruvate. Okay, cysteine can then go to pyruvate, like we saw a couple slides ago. Okay, all right. Now I think this is where we stopped last time. Amino acids degrading to acetyl-CoA. Okay, and acetyl-CoA. Okay, acetyl-CoA is a central metabolite that is used in TCA cycle, that is used in fatty acid biosynthesis. Right. So right off the bat, all of the branch chain amino acids, like what we saw with leucine, isoleucine, of course, they degrade to acetyl-CoA, right? So we saw that the branch chain amino acid degradation in about 3 slides ago, they degrade via a beta oxidation-like pathway, okay? So if they do a beta oxidation-like pathway, then, of course, they generate acetyl-CoA. 可以哈，懂我意思哈？就是我们刚刚看到 branch chain amino acid 那些 pathway 有没有？我们不是说会有3个 process， 第一个 transamination， 我先把那个 NH2 弄不见，然后第二个我做一个 oxidative decarboxylation， 把那个 alpha keto acid 的那个 acid 去掉 ，OK， 然后 alpha keto acid 会 become 一个 Acetyl-CoA，OK， 那 Acetyl-CoA 接下来就是做 beta oxidation 的 pathway 类似的 reaction，OK， 那 beta oxidation pathway 类似的 reaction 给你的就是 acetyl-CoA, right? 所以 no surprise that these branch chain amino acid are used to give you acetyl-CoA. Okay, make sense? Right? Now, turns out, so leucine, isoleucine, that's fine, right? Turns out, tyrosine, phenylalanine, and lysine, they also end up at acetyl-CoA. Okay? Now, we're going to take a look at the pathways associated with phenylalanine tyrosine and lysine. Okay? But again, okay, you don't have to memorize the pathways. You just have to know the key chemical reactions. Okay? So, 那种 transamination 啊，或者是 hydroxylation 这种东西你需要知道。Okay? Lastly, we have tryptophan. Okay, so these 3 are the last amino acids that, these 4 are the last amino acids that we're going to talk about. Okay? So you have a total of 7 different amino acids that can degrade into acetyl-CoA. They are phenylalanine, tyrosine, lysine, tryptophan, leucine, isoleucine. Okay? And threonine. Threonine we saw in the previous slide. Now, lysine degradation. Okay? Lysine degradation is going to be a very interesting process because if you look at lysine, okay, you have 1 carbon, 2, 3, 4, 5, 6. You have 6 carbons, okay? And it's an amino acid with a terminal omega amino group, okay? Now, if you look at this, okay, without using this pathway, okay, without looking at this pathway, how would you degrade lysine? 就是说，如果你先不要看，假装你没看到这个 saccharopine 的这个 intermediate， 如果我说你今天就是 lysine， OK， 然后 lysine 你有2个 amino group， OK， 然后我今天要把 lysine degrade 掉 ，OK， 你觉得如果今天是你们的话，要用什么样子的 chemical reaction 可以把 lysine degrade 掉？第一步是什么？我们做 amino acid degradation 的第一步大概会是什么 ？transamination， 对不对 ？Get rid of 我的 amino group，right？ So 我可能可以先把我的 amino group get rid of， 这是一个 transamination 我可以做。这边我也可以做一个 transamination，right？Which is true, right? So if you do a transamination of the omega amino group, doesn't it just become this? It becomes an aldehyde, okay? So your intuition is actually right? So essentially, you want to convert this, which is an amino group, into this, which is a aldehyde group, okay? So in a way, this is a transamination, okay? How do I know that? If you take a look at the saccharopene intermediate, you throw in alpha Kg, you end up glutamate, okay? Here you consume NADPH, but here you produce NADH. So redox is neutral, right? 一边我吃一个 NADPH, 但另外一边我吐一个 NADH. Even though 你有那个NADP 跟 NAD 的 difference, 但是站在一个 energy point of view, 它们就是 redox power, 对不对？就是 reducing agent 嘛。OK， 所以一边是吃 reducing agent， 然后另外一边吐一个 reducing agent， 所以这边的话 electron 是 balanced 的，但是the net reaction 就是你 convert 了一个 alpha Kg 到 glutamate，OK， 然后你把你的 lysine 变成这个 alpha amino adipic semialdehyde，OK， now what would be next? Okay, so aldehydes in general, okay, you guys should always remember this. Aldehydes in general, not good for your body, okay, so you want to get rid of aldehydes, right? So obviously, what are you going to do? Okay, 有没有知道你们像如果你今天喝酒精嘛，喝酒， okay, 酒精的代谢是怎么代谢？酒会先变成就 ethanol 会变成什么 ？acetaldehyde， 对不对？乙醇变乙醛嘛 ，OK， 然后 acetaldehyde 之后 ，OK 会变成 acetic acid， 然后变成 Acetyl-CoA，right？ 所以站在一个代谢的 angle， 我们都会把不同的 aldehyde 变成 acid。Okay, so what we're going to do is we're going to oxidize the aldehyde into an acid, and then we're going to transaminate this amino group into keto group, such that you end up with this thing called the alpha keto adipate. Okay, alpha keto adipate. Adipate is a 6-carbon diacid, and you got this alpha keto, therefore this guy is alpha keto adipate. Okay, now alpha keto adipate 呢，它其实跟那个 alpha keto glutarate 长得很像。alpha Kg 它是5个碳 ，alpha keto adipate 是6个碳，就这样而已。Okay, right? Now alpha keto adipate can then degrade into acetyl-CoA. Okay. Tryptophan degradation. Okay. Now, tryptophan degradation, you take a look at this thing and you'll be like, whoa, what? Okay, it looks kind of crazy. Yeah, it does look kind of crazy. All right, now, you start with tryptophan, okay? And tryptophan has this aromatic portion, and you got these 1, 2, 3 carbons, okay? So what are we going to do, right? Now, turns out the first thing we're going to do is going to somehow break the aromaticity or break the ring structure, right? So we're going to use an enzyme called the tryptophan 2,3-dioxygenase. Okay, now, dioxygenase, we previously saw monooxygenase. Okay, monooxygenase uses oxygen, and you only put one oxygen atom into your molecule. Dioxygenase incorporates both of the O2. Okay, O2 不是2个 O 吗？ Dioxygenase 呢，它就是会把2个那个 oxygen atom 都塞在你的 molecule 身上。Okay? Now, so this is what you would do. Right? You would take your tryptophan 2,3-dioxygenase and you're going to put one oxygen right here, one oxygen right there. Okay? So essentially you react it with the gamma carbon. Okay? So this I'm going to erase all this. This is the gamma carbon, and we're going to react it with these two. Okay, so one O goes to here, the other O goes to here. Okay? And then you will generate your N-formyl quinoline. Whatever, you don't have to memorize the structure, okay? This reaction, you don't have to remember, okay? But I do want you to understand what is a dioxygenase, okay? 但是这个 particular reaction 你不用去记它， okay? 因为这个不太好记，因为你有 ring breaking 嘛， okay? Now, so going from this, what you can do is you can hydrolyze a formic acid out. So 如果你把这整个切掉 this is formate. Basically this. Okay? And then you have another, guess what? Oxygenase. Okay? Because it uses oxygen. Okay? And you incorporate a hydroxy group onto the phenyl ring. Okay? Or oxygenase, or I think usually we'll call them hydroxylase. Okay? But it's just a type of oxygenase. Hydroxylase, you put a hydroxyl group on it. Okay? Now, after that, you can cut it. You can cut this molecule in half, where you end up with alanine, and you end up with this 3-hydroxyanthenic acid, which then, okay, you use another oxygen to break the ring structure. Okay? So you're going to break the ring structure such that you generate this weird aldehyde looking thing. Okay? And now you have two routes. One route is where in many steps you can get to alpha ketoadipate, and then we can go to acetyl-CoA just like the previous slide. Or you can use this guy to then generate or synthesize the NAD and NADP. Okay? So turns out tryptophan is an important precursor to NAD biosynthesis. Okay? But again, this slide, you don't have to remember the chemical structures. Okay? Just understand what is an oxygenase. All right? Now, for tryptophan degradation, what you should understand is that you see in the entire pathway, it uses many oxygen as your co-reactants. Okay? So this degradation process occurs under aerobic condition. Okay? So for the bacteria that's anaerobic, they will not use this pathway. 可以吧？可以哈？ Right. All right, lastly, we have phenylalanine and tyrosine. Again, phenylalanine has a benzene ring right here, and tyrosine has a phenol ring right here. Okay? So turns out, if you want to degrade phenylalanine, you're going to turn phenylalanine into tyrosine. Okay, huh? Phenylalanine into tyrosine. Now, this reaction you have to remember, it's called the phenylalanine hydroxylase. Hydroxylase, in a way, it's kind of like it's a type of oxygenase because it uses O2 as the reactant. Okay, it uses molecular oxygen as your substrate. Okay, it's called hydroxylase because it introduces a hydroxyl group. Right? Now, as we know in chemistry, benzene rings are in general unstable, 对不对? Aromatic things 非常非常 stable. 你要 introduce chemical functional groups 在东西上面其实不是很容易的一件事情，合理吧? Okay, so the ability for this guy to put an OH onto the benzene ring is actually a very powerful tool. Okay, now, so it turns out you're going to throw your hydroxyl group onto your benzene ring, forming a phenol. Then what you can do is transamination. Okay, so this guy is transamination. Okay, and then you have a dioxygenase, which you will use to do a very crazy reaction where essentially what you have done is you move the hydroxyl group around and you got rid of the keto group. Okay? Then subsequently you will use another dioxygenase, okay, which then you would be able to break the aromatic ring such that you generate your linear compound. malonyl acetyl-acetate, which then you do an isomerase reaction, okay, whereby you change the structure from this is the stereochemistry of this guy. The two hydrogens are here. Now they are on the opposite side. Okay, so it's, what do you call those? Cis-trans isomerization. Okay. Okay, huh? Right. Subsequently, then what you would do is cut the molecule in half, and you generate fumarate, and you generate acetyl-CoA. And acetyl-CoA goes to where? Acetyl-CoA. OK， 因为这是 beta oxidation 的最后一步嘛 ，acetyl-CoA 到 acetyl-CoA。OK， 所以你的那个 phenylalanine tyrosine when it degrades into fumarate and in this case acetyl-CoA. So therefore, these guys, they're both ketogenic and glucogenic. Make sense? Because the products that they produce can both be used to generate ketone bodies and be used to generate glucose. Right? Fumarate can go to oxaloacetate. Oxaloacetate can go to pyruvate. What do you notice on this slide is a bunch of these X's. Okay? So it turns out, okay, there's a lot of hereditary diseases, okay, 那个遗传性的那种疾病啊，或是 genetic disorder associated with the malfunctioning of these enzymes. Okay, look, 你们有没有习惯吃代糖 ？Okay, sugar substitute. 应该可能在台湾不是那么流行啊，但在美国非常非常流行。就是你去那个咖啡厅有没有？它那个旁边不是都会有放那个 sugar 吗？那不是会有那什么 brown sugar， 然后什么一般的 white sugar， 然后有一堆不同的代糖。OK， 那其中一种代糖呢叫 aspartame。OK， 这个东西叫 aspartame， 这是一个 sugar substitute。OK， now this sugar substitute is composed of three things: aspartic acid, phenylalanine 跟 methanol。OK， 那这个 sugar substitute 呢很 interesting 哦，因为它长得跟 sugar 完全不一样嘛。一般来说 ，sugar 都是 carbohydrates， 那这不是 carbohydrates，OK， 所以这个东西就是它虽然吃起来会有一些甜味，但它不是 sugar， 所以说我们 degrade 这个东西的时候，它的degradation pathway 呢，比较不太一样，它会进入到变成 aspartic acid 跟 phenylalanine。So when you degrade this molecule, what you get is aspartate, phenylalanine, and then methanol. OK， 那被人家最 criticize 的其实是这个 methanol，right？ Like 你总不会跟我说 methanol 喝下去应该没事吧？不 OK 嘛 ？methanol 就是那个假酒有没有？就是喝了眼睛会瞎嘛。当然了，那个糖这个 dose 不是很高啦，所以是还好。But it's still not very good. Okay, now this is what normal people would say. Now, in particular, a type of group of people are not suitable to take aspartame. Okay, 那这些人呢，就是他们有一些genetic defects 在 phenylalanine 跟这个 tyrosine 的 degradation 上。Okay， 那如果他们今天吃代糖，对不对？那你代糖它会被水解成 aspartate 跟 phenylalanine 跟 methanol。那你没有办法去 degrade phenylalanine。Okay， 那 as a result 你会 actually accumulate phenylalanine in your body. Okay, so it turns out you actually cause mental retardation, that kind of stuff. Right? So it's actually a very serious problem. Right? So a lot of times babies are tested for these type of genetic diseases. Okay, now. So this is the last slide for amino acid degradation. We just wanted to show you that human genetic disorders associated with amino acid metabolism. Okay, so you got a bunch of these. Right? So the thing that I was just talking about was this phenylketonuria. Okay, it lacks phenylalanine hydroxylase, so it cannot metabolize phenylalanine. So as a result, you get mental retardation. You get vomiting, that kind of stuff. Okay, so it's not comfortable. You also got some other stuff. For example, albinism. Okay, 这个有没有听过 ？albino 有没有听过？就是你那个缺少合成那个色素的 genes， 所以人会看起来很白，有没有？什么眉毛啊，全部都是白的。 那这个东西呢，主要是因为他们缺少，他们 defect 一个 enzyme 在这个 tyrosine 3 monooxygenase。OK， 那 as a result， 它没有办法去做这个 melanin。melanin 我们 hopefully 那个下礼拜会讲到。OK， 怎么样子合成你的那个这些不同的这种奇怪的 compound。OK，melanin。Right? And you also got other stuff. So you can take a look at this chart. So you can see that a lot of these enzymes are all related in amino acid metabolism. So typically, if you have mutations in amino acid metabolism, what you usually end up is some kind of mental retardation, typically. Okay, so for human genetic disorders, the most common end result is usually some kind of mental problem. Okay, so now maybe somebody somewhere will figure something out to fix that. Okay? All right, so this is amino acid degradation and urea cycle. Okay, so what do you have to know? Urea cycle, you have to know. Okay, you have to know urea cycle. And for amino acid degradation, what do you have to know? Okay, you have to know some common reactions. Okay, what do I mean by common reactions? Hydroxylases, that kind of stuff you have to know. For example, how do you go from benzene ring to somewhere in that has a OH on it? Okay, hydroxylase. You have to know that. You have to know transaminase. Okay, you have to know decarboxylase. Okay, amino acid decarboxylase. That you have to know. Amino acid racemase. Okay, so PLP dependent 的3个 enzyme 我们不是有讲，我们有讲 transaminase、racemase 跟 decarboxylase。这3个麻烦一定要知道。OK， 所以我可能可以问你一个问题说 ，OK， 在 E. coli for example， 他们会去做 D-alanine。OK， 那你可能要告诉我说，那 D-alanine 是怎么来的？那从中间你可能就会用到一个 racemase。OK， 就是你要知道这样子 level 的一个 understanding 就好。OK， 那像这种什么 phenylalanine 啊、tryptophan 这种 degradation， 这 pathway 真的一点都不重要。OK， 就是只是跟你讲说 they exist and this is what they look like。Okay, but you know it's absolutely not important for you guys to memorize that. Okay? But I do want you guys to know the common simple reactions. 这种 transamination 啊，怎么样子把不同的 functional group 做修饰、做改变。Okay, 这个要知道。Okay, 因为这个对于metabolism 来说还蛮重要的。